to find a discovery of this nature, was certainly nothing that we expected.
A 14,000-year-old skeleton from an Alaskan cave was supposed to confirm what every textbook said about how the first people reached the Americas. Instead, when its DNA was finally read, it said something nobody was prepared for. These bones spent 60 years in a museum drawer, ignored because the science to question them did not exist yet.
We've got the presence of two infants that are buried in a pit. So a pit was dug out, the infants were laid there.
Now they do not just challenge the migration theory we were all taught. They quietly rewrite it, and the strangest part is who these people actually turned out to be once the genome could finally speak. A cave between continents. The cave sits on the western Alaskan peninsula that juts toward Russia across the Bering Strait, cut into hills above a dry tributary of the Imuruk Basin. The site is called Trail Creek Caves, and for understanding how the Americas were first populated, it is one of the most consequential single archaeological sites anywhere in the Western Hemisphere. Larson did not know that. He was working in a remote cave above a creek bed, recovering what he could before the season ended. The human bone fragments came up alongside the remains of mammoth, bison, and caribou, the animals of the late Pleistocene tundra, the cold open grassland that surrounded the cave when its first human occupants were alive. Then the bones did what bones excavated in that era almost always did. They went into a museum collection in Denmark and they stayed there, cataloged, stored, largely untouched. The science that could make them speak did not exist yet, and would not for decades. In 2010, the bones came home. Under the Native American Graves Protection and Repatriation Act, the Trail Creek Caves remains were carried back to Alaska. It was, on paper, a routine administrative matter, a transfer of custody under a federal law. Nobody loading those boxes expected them to overturn anything, but once they were back, they were radiocarbon dated, examined morphologically, and made available for the kind of analysis that had been impossible when Larson first pulled them from the dirt. And in the decade that followed, as techniques for recovering ancient DNA improved by orders of magnitude, the genetic material locked inside those fragments finally became something a laboratory could read. What it said did not match the story in the textbooks, not the details, not the broad strokes. To understand why a single cave on the Seward Peninsula could break a chapter of American prehistory, you have to understand the model it broke first. The model that ruled. For most of the 20th century, mainstream archaeology had one tidy answer to how the Americas were peopled. One migration. A population of big game hunters crossed the Bering Land Bridge from Asia roughly 13,000 years ago, then spread rapidly across two continents, becoming the ancestors of every indigenous American population from the Arctic to Tierra del Fuego. The model was simple, it was elegant, it was easy to teach, and it was wrong. It even had a name and a founding artifact. In 1932, near the town of Clovis, New Mexico, archaeologists found a distinctive style of fluted stone projectile point lying among the bones of mammoths. The points were beautifully made, large and leaf-shaped, with a channel flaked up from the base on both faces. Nothing quite like them had ever turned up in the Old World. When radiocarbon dating became available in the 1950s, it placed the Clovis culture at roughly 13,000 years ago. For the next half-century, those points were treated as the floor of American prehistory, the earliest unambiguous evidence of humans in the Americas. The people who made them were assumed to be the first Americans, hunters who walked from Siberia to Alaska when low sea levels exposed the continental shelf, then traveled south through an ice-free corridor that opened between two great ice sheets as the glaciers pulled back. One founding population, one migration, one technology sweeping two continents. Clovis First was not just a theory, it was orthodoxy, and it was defended like one. Any claim of people here before Clovis met intense skepticism. Sites that looked older were attacked on methodological grounds. Researchers who pushed for earlier dates often found their careers quietly marginalized. The consensus was enforced not only through scientific argument, but through institutional pressure, through which papers got published, which grants got funded, which excavations were taken seriously. For decades, the safest career move in American archaeology was to find nothing older than Clovis. That is not a neutral scientific environment. That is a system that rewards one answer and punishes the rest, which is a big part of why the model survived as long as it did even after the cracks started showing. Before we get to the sites that refuse to die quietly, here is the deal. If following the evidence past the version you were taught in school is the kind of thing you want more of, subscribe, because that is the entire point of this channel. Now, back to the cracks in the model. The sites that refused Monte Verde in southern Chile produced evidence of human occupation around 14,500 years ago, more than 1,000 years before Clovis. The site preserved wooden structures, stone tools, and plant remains inside a peat bog that had shielded them from decay. Here is why that was so hard to dismiss. Organic material almost never survives. Here it had, and it described a settled camp, not a quick passage through. The dates were challenged, they were defended. More excavation confirmed them. And the location did the real damage because Monte Verde sits near the southern tip of South America, not just inside Alaska. If people were that far south that early, they had to have entered the hemisphere much, much earlier still. Meadowcroft Rock Shelter in Pennsylvania produced dates potentially reaching back 16,000 years or more. Critics argued ancient carbon might have leached through the rock and poisoned the lower layers. The excavators answered every objection, and the deep deposits held. The Buttermilk Creek complex at the Friedkin site in Texas yielded thousands of stone tools in a clean layer beneath the Clovis deposits, with ages approaching 15,500 years. The stratigraphy was tight. The artifacts were unmistakably made by human hands. Each site faced intense scrutiny. Each one survived it. Any single one, on its own, you could argue about over coffee for years. But 3 of them, on different parts of 2 continents, all pointing the same direction, is not an argument anymore. It is a pattern. And a pattern is the one thing a defended orthodoxy cannot survive. The pre-Clovis evidence was real, and it was accumulating. But the decisive break did not come from archaeology at all. Ancient DNA changed everything. It came from genetics, and at first it almost did not work. The technology for pulling human DNA out of old bone was refined mostly in Europe, where cold and favorable burial can preserve genetic material for tens of thousands of years. The Americas were harder, warmer climates, different burial practices, acidic soils that chew DNA apart fast. And here is the thing people miss about ancient DNA. It does not survive as a clean strand you can just read off. It survives as a haystack of short, broken, chemically scrambled fragments, contaminated by the DNA of everything that has touched the bone since the person died, including the modern hands holding it. Reading an ancient genome means reassembling a shredded document where most of the pieces are missing, and a lot of the surviving ones are forgeries. But the techniques kept improving. The amount of material needed kept dropping. By the mid-2000s, researchers were sequencing genomes from skeletal remains all across the hemisphere. And the first results both confirmed old ideas and immediately wrecked them. The Anzac child, an infant burial from Montana, found with Clovis artifacts, was sequenced in 2014. The genome showed Clovis people were ancestral to modern Native Americans, and that they already represented a population diversified from the original founding line. Already diversified. That word matters more than it looks. It means by the time the Clovis people existed, the splitting had already happened. They were not the start of the story. They were a chapter somewhere in the middle of one nobody had read yet. The Kennewick Man, a roughly 9,000-year-old skeleton from Washington state that had triggered years of legal fighting over whether he was Native American, was sequenced in 2015. The answer was not ambiguous. He was closely related to modern Native American populations of the Pacific Northwest. The Spirit Cave individual from Nevada, around 10,600 years old, was sequenced in 2018, confirmed as Native American, and repatriated to the Fallon Paiute Shoshone tribe. These results locked in the method and confirmed the framework. The people living here at the end of the Pleistocene were ancestral to the indigenous peoples who were here when Europeans arrived. But the same data delivered something nobody asked for. The ancient Americans were not one homogeneous population, spreading evenly across 2 continents. They were multiple genetically distinct groups, split from each other thousands of years before Clovis, taking different routes, holding different regions, leaving different signatures in their descendants. The single migration model was not bending, it was breaking. The infants that rewrote the decisive find came out of central Alaska. Picture Ben Potter, an archaeologist from the University of Alaska Fairbanks, in 2013, standing over an excavation in the Tanana River Valley at a site called Upward Sun River. The deposits date to roughly 11,500 years ago, after Clovis, but among the oldest human occupations anywhere in Alaska. Beneath a layer of burned material in a residential pit, his team uncovers the remains of 2 infants. The preservation is extraordinary. The burial is undisturbed. He is looking at the oldest human remains ever found that far north, and they are children. The remains went to a team led by Eska Willerslev at the University of Copenhagen. In his lab, technicians did the painstaking work of pulling that shredded, contaminated genetic material out of the bone and reassembling it, fragment by fragment, into something a person could actually read. The results, published in Nature in 2018, rewrote the peopling of the Americas. That is the first time the word fits, and it fits exactly. One infant, designated USR1, yielded a complete genome. It did not match any known Native American population. It did not match any of them. It was a separate lineage that had split from the ancestors of all other Native Americans roughly 20,000 years ago, and then stayed isolated in the far north for thousands of years. They named this population the ancient Beringians. Read that again. The ancient Beringians were not the ancestors of most modern Native American groups. They were a parallel branch, a population that broke off from the main founding line while both were still up north, before the migration south even began. So, the peopling of the Americas was not one wave, it was at least 2. The ones who stayed and the ones who kept going. And that was before the full complexity emerged. The Trail Creek Bones. Now, Larson's bones walk back into the story. The Trail Creek Caves remains were run as comparative material for the Upward Sun River work and everything that came after it. And they confirmed the Beringian complexity. The people buried in those limestone caves on the Seward Peninsula belonged to the same deep population structure the infant genome had exposed. Think about what that actually means in time. Those fragments came out of the ground in the late 1940s, before ancient DNA was even a concept, while Clovis I was still hardening into law. They sat in a drawer in Denmark through the entire era when the single migration story was taught as settled fact in every classroom on the continent. They were carried back to Alaska in 2010 as a paperwork matter, with nobody expecting anything. And when the science finally caught up to them, they turned out to be a physical witness to the exact thing the textbooks swore could not have happened. They were a snapshot of the genetic diversity of late Pleistocene Beringia, a diversity the single migration model never predicted and could not explain. The same bones Larson wrapped without knowing what they were. They did not just fit the new picture, they are the new picture, holding still, waiting 60 years for someone to look. They also helped firm up another idea, one argued on theory long before genetics could test it: the Beringian standstill, a drowned land. The idea, developed by researchers including John Hoffaker of the University of Colorado, goes like this. The ancestors of Native Americans did not sprint from Siberia into the Americas in one continuous push. They stopped. For thousands of years, they lived in Beringia itself, the exposed continental shelf that joined Asia and North America when sea levels were hundreds of feet lower than today. Not a tense pause at a border crossing. Generations. People being born, living entire lives, and dying in a place they never left, while the route ahead stayed sealed behind a wall of ice. And here is the part that gets me. Beringia was not just a bridge you crossed. It was a place you lived, a refugium, a habitable pocket where people could survive through the worst of the Ice Age while massive ice sheets buried Canada and sealed off any route south. The genetics put numbers on it. The ancestral population sat isolated in Beringia for as long as 8,000 years, from roughly 25,000 years ago, when they split from their Asian source, to around 17,000 to 15,000 years ago, when they finally moved south as the ice retreated. 8,000 years. That is not a layover. That is longer than all of recorded human history. That is enough time for a population to drift into something genetically distinct, to become its own people, separate from who they left in Asia and who they would become here. And it was not one quiet group waiting patiently to move. It was a population splitting and branching in place. The ancient Beringians peeled off from the line that would become northern and southern Native Americans. Other groups may have emerged in that isolation too, lineages we have not sampled, lineages that may have ended with no descendants at all. We will never get most of them. The land they lived on is at the bottom of the Bering Strait. The standstill happened in a country that no longer exists, and the people in it left almost nothing we can reach. The ghost in the genome. In 2015, a study led by Pontus Skoglund and David Reich found something stranger still. Some indigenous populations of South America, particularly groups in the Amazon basin, carried a small but statistically real genetic component that looked like it came from a population related to Australasians, the indigenous peoples of Australia, New Guinea, and the Andaman Islands. This component, called Population Y, did not show up in most Native American groups, only certain South American ones, and it had no obvious source. Australasians were never proposed as contributors to the peopling of the Americas. The distances were enormous, the routes made no sense, the timing was a problem, and the signal was still there. The best explanation was a ghost population, and the word ghost is precise here. It does not mean imaginary, it means the opposite. This population was real enough to leave its fingerprints inside the genomes of living people, but it left no living members of its own. No group anywhere on Earth descends from it directly. It exists now only as a statistical shadow inside other people's DNA. Maybe it arrived separately. Maybe it was absorbed by later arrivals. Maybe it was wiped out by them. Whatever happened, the only thing left of it is a faint Australasian echo in a handful of South American groups and silence everywhere else. The peopling of the Americas was not one migration. It was not even two. It was a process, a tangled multi-wave, multi-source process running over thousands of years, involving populations we are only now starting to see. The footprints that broke. Then the timeline itself snapped. In 2021, a team led by Matthew Bennett published evidence of human footprints preserved in the sediments of White Sands, New Mexico. These were not artifacts you could argue away on typology, not animal bones you could call natural deposits. They were footprints, human feet, pressed into the mud of an ancient lakeshore, unmistakable. And they dated to roughly 21,000 to 23,000 years ago. The dating was attacked. It relied on radiocarbon ages from aquatic plant seeds, which can soak up old carbon from water and read older than they are. So, in 2023, a follow-up by Jeffrey Pagady, Kathleen Springer, and colleagues reconfirmed the ages with multiple independent methods. The footprints were real. The dates were real. Humans were in the interior of North America at the height of the last glacial maximum. Sit with what that breaks. Thousands of years before the ice-free corridor opened. Thousands of years before the standstill was supposed to have ended. Thousands of years before anyone was supposed to be there at all. If those dates hold, people reached the middle of North America while ice still buried Canada, and the corridor route simply did not exist yet. That is not a small revision. That is the whole timeline coming off its hinges. A coastal route down the Pacific, sometimes called the Kelp Highway, might have been open with people moving south by boat and on foot along a shoreline thick with food. But the timing pushed the very edge of what that idea predicts. And the genetics did not cleanly account for anyone here that early, and footprints carry no DNA. They tell us someone walked here. They do not tell us who or where that lineage went, or whether anyone alive carries a trace of them. Were the White Sands people related to the ancient Beringians, an even earlier unknown population that left no descendants, the source of that ghostly Australasian signal? Did their line just end, leaving nothing behind but tracks in a lake bed for 20,000 years? The questions multiplied faster than the answers. A research frontier. Jennifer Raff, an anthropologist at the University of Kansas and author of Origin, A Genetic History of the Americas, has tried to pull the current picture together, and her honest assessment is not comforting. The simple stories are gone. The textbook diagram with one arrow from Siberia to the Americas is obsolete. What is left is a frontier. A field where new data arrives faster than agreement can form, where every ancient genome hands you answers and a fresh pile of questions in the same breath, where the story is being rewritten while we watch. The Trail Creek Caves remains are part of that rewriting. there is the word again, and it is not decoration, it is literal. The chapter is being edited in real time, and these bones are one of the edits. They are not unique, they are not even the most dramatic find of the past decade, but they are representative. They show what Beringia looked like genetically at the end of the Pleistocene, a population split into multiple lineages, some becoming modern Native Americans, some staying in Alaska, some ending entirely. And they raise a question that should bother you a little. The Trail Creek bones spent 60 years in a Danish drawer before anyone could read them. How many other collections are sitting on the same kind of data right now? How many skeletons pulled out of the ground in the 19th and 20th centuries, before ancient DNA was even imaginable, are holding genomes that could settle questions we are still fighting over? Nobody knows. That is the unsettling part. The revolution is not winding down. New methods pull signal from samples that used to be hopeless. New collaborations between geneticists and indigenous communities are building the ethical frameworks this work demands. The next decade will likely produce more data than the previous century, and every bit of it is going to complicate the simple stories further before it ever simplifies anything. The bones finally speak. Clovis, first, is dead. The single migration model is dead. The ice-free corridor as the one road in is dead. What is left is complexity. The recognition that the peopling of the Americas was as complicated as the peopling of any other continent, that multiple populations contributed, that the process ran across tens of thousands of years, and that we have only started to understand what actually happened. Most of the original land bridge is gone, drowned under the Bering Strait as the seas rose. Whatever sites once sat on that exposed shelf, the camps, the kill sites, the burials from the thousands of years people lived in Beringia, are underwater and out of reach. The coastal route, if it was used, left most of its evidence on shorelines hundreds of feet below the Pacific. The deepest history of how the Americas were peopled is, to a real extent, simply gone. What survives are the margins. Sites like Trail Creek Caves, sitting above the old coastline, where the evidence still exists in a form we can actually touch. They are rare, they are precious, and the work is nowhere near done. Researchers including Dennis O'Rourke and Connie Mulligan, alongside Raff and others, keep pulling ancient DNA out of Beringian and early American remains. Each genome adds to the pile. Each one reveals a new pattern, or confirms an old one. The picture coming into focus is not simple, but it is true. The ancient Beringians were real. The pre-Clovis sites were real. The ghost with Australasian affinities was real. The White Sands footprints were real. The simple stories, the ones taught in school, were incomplete at best and wrong at worst. The new one is still being written in the language of ancient DNA, pulled from bones that waited decades or centuries to give up what they were holding. The same bones Helge Larsen lifted out of a cave floor with his bare hands, not knowing they would one day rewrite the textbook he learned from. The caves are still there, cut into the limestone hills above the Imuric Basin. The bones have spoken. Somewhere in a museum drawer, a lab refrigerator, an archive nobody has opened in years, the next genome is waiting to be sequenced, ready to add another chapter to the longest migration in human history. So here is the question worth sitting with. If a few forgotten bones in a Danish drawer could overturn a century of consensus, what else is sitting in storage right now, waiting to rewrite a chapter we think is already closed? Tell me in the comments which part of this surprised you most. Subscribe so you do not miss the next one, because the next investigation goes straight into the White Sands footprints and the people who left them, thousands of years before anyone was supposed to be there.